So we continue with respiration, and as I told you, this particular video will be based on only the experiments which are given over here. There are totally 5 experiments for proving the different kinds of things what are taking place during respiration. So uh, the first one is experiment to prove that oxygen is used up in respiration. So for this particular condition, what we have got is we have taken two flask. We have taken 2 flasks, one flask here and another flask over here, and into the flask you have got certain wet cotton, and wet cotton के ऊपर हम लोग ने डाला है कुछ seeds. So this seeds, and the seeds over here also. But only difference is that these are dead because we have boiled it. So these are boiled, so dead, whereas these are not dead. So these are normal, which can germinate. So germinating, so these are germinating. Okay, this is cotton, wet cotton. And what we do is we are connecting it to a tube. Now, you will see that in any experiment of biology, we will be having 2 setups. In one of the setup, we are sure that nothing is going to happen. Like यहाँ पे dead body है, dead seeds है. Now we know that if it is dead seeds, definitely they are not going to respire. So there should not be any change over here. So this is called as the control setup. Whereas this over here is the experimental setup. So the one in which you are expecting the changes is the experimental setup. The one in you are sure that nothing is going to change is the control setup. Now why do you need a control setup? Just prove that अगर ये नहीं होता तो मैंने अदरवाइज नहीं तो नॉर्मल है. This happens even if you don't put a seed over here. ऐसा नहीं है. If you don't put a seed or you put dead seeds over there, then such a thing is not going to happen. So that is why it is called as a control setup. You are going to put a trough of water over here. So definitely there will be water coming up to this point over here initially, both of them coming over here because of the capillary action. Now, what we are doing is we are placing over here a test tube containing soda lime, and over here also we are taking a test tube containing soda lime. So we are going to have soda lime on both the sides of the test tube. Now what is going to happen is this particular thing is going to respire, and whatever oxygen is going to be given out. But if see here there is air right now. Okay, there is air right now. And this air which is there. Now what we have done is we are using the soda lime. We have dissolved the carbon dioxide which was there in this particular container. Soda lime is absorbing the carbon dioxide, so that is the point. So we are absorbing the carbon dioxide. तो अभी क्या बचा है यहाँ पे? Oxygen बचा है, nitrogen बचा है. Nitrogen तो लेता नहीं है. So you will find that यहाँ पे there is no change, whereas this one, because the oxygen is being utilized by the plants for germination, that's why the content of the air inside over here will decrease. That's why there will be low pressure generated, because whatever carbon dioxide is going to be given out, that is going to be absorbed by the soda lime. So here oxygen is taken in, carbon dioxide which is given out is absorbed by soda lime. So there will be a depression, there will be over here vacuum formation. Accordingly, ये water level rise होगा. So the rise in the water level shows that oxygen was utilized for photosynthesis, for respiration. So let's see. An apparatus is arranged as shown. Figure flask A contains germinating bean seeds and flask B as dead boiled seeds together with some antiseptic. Now why do you need an antiseptic over there? Because you don't want that the boiled seed or dead seeds which are here, they get some microorganisms on it, or basically some bacteria on it, because if the bacteria growth is there, then they are going to respire and we will feel that ये dead plant भी respire कर रहा है. Right? That's why we don't want any kind of microbial growth on the dead plant, dead seeds. That's why we are putting some antiseptic over here. Wet cotton provide water to both samples. A small test tube containing soda lime, a mixture of sodium hydroxide and calcium oxide, is suspended in each flask for absorbing any carbon dioxide released by seeds. Okay, that is the reason we are using soda lime. So soda lime is going to absorb the carbon dioxide which is being released or which is present in the air already. After a few days, the delivery tube connected to the flask A will show a greater level rise in the water level. When the burning paper is introduced in each flask after removal, their corks, the flame is immediately put off in A, but it is continuous for a short while in B. This proves that oxygen was absent in the flask A. So what we see over here, first thing is that there is a level difference over here. Secondly, if we bring a glowing burning candle over here and a burning candle over here after a few days, you will find this will immediately get extinguished because it requires oxygen for its burning, but there is no oxygen available here, so it extinguishes very immediately, whereas this one will extinguish after some time because this one has got oxygen balance in it. This proves that oxygen was absent in flask A, thereby showing that it was used up by germinating seeds, and the volume of oxygen so used up was indicated by the rise in the water level. Okay, the oxygen यहाँ पे इतना जितना use हुआ, that was seen by the rise in the water level in the delivery tube. There are two simple questions which you can surely answer. Why is there a slight increase in the level in the delivery tube of B? So, and which is of the two flask is a control setup? Of course, this is the control setup and this is the experimental setup. And they are given a slight increase which is there. So maybe that is because of carbon dioxide which is present inside the air is absorbed by the soda lime. So वो soda lime absorb कर लेगा तो थोड़ा बहुत. Anyway, don't worry about that. Such questions are not so important. Okay. So a proper experiment must have two components, experimental and control setup. An experiment consists of two setups, an experimental setup and a control setup. Both are identical in which every condition is the same except one. The setup in which the condition under study is missing is called as a control. So here the study is for the plants, the seeds are going to take in oxygen, respire. We know dead will not respire. So that's why this is the control and this is the experimental setup. We now see the next experiment, that is to prove that carbon dioxide is produced during respiration. So the next experiment is to prove that carbon dioxide is going to be released while respiration, and so we are going to have it in two ways. One is the respiration by seeds and the other one will be respiration by plants. So the second experiment will be about seeds. So it's a simple thing. What we are taking is we are taking a flask. वो फ्लास्क दो फ्लास्क लिया हम लोग ने. दो फ्लास्क के अंदर of course as usual cotton and seeds. एक के अंदर boiled seeds and dead, that is the control setup, and एक के अंदर germinating seeds. You kept it for some time. So the next what you do is you are going to open that particular thing after some time and you are going to take a lime water solution. Okay, we know that lime water, when carbon dioxide passes through lime water, it turns milky. Now carbon dioxide is heavier, so what we are going to do is just you are going to take that lime water and वो lime water और ये flask लिया, flask में से हम लोग ऐसे जैसे pour कर रहे हैं. We cannot see it, but we are pouring it. When you are pouring it, what happens? Carbon dioxide would enter downwards. So this will show that oxygen और जो respiration हुआ तो उससे carbon dioxide generate हुआ और वो lime water को milky turn किया. So it will be like a magic for you because you will be having the test tube over here which will be having lime water which will be transparent, which will not be milky, it will be transparent. And when you take that particular, suppose this is the flask, I am taking the flask, I cannot see anything over here, but when I pour it like this, so we will feel that after some time this will turn milky. तो उसमें उसका मतलब क्या हुआ कि gas, the carbon dioxide which was invisible because it was transparent, we cannot see that carbon dioxide, it entered over here and it turned the lime water milky. So this is what is there in experiment 2, that take two flasks A and B, place some cotton wool on at the bottom of each flask, soak seeds such as a bean seed are placed in flask A and equal number of boiled seeds or dead seeds are placed in flask B. Antiseptic or such as carboic acid is added to flask B to prevent bacterial growth of dead seeds which would otherwise respire and release carbon dioxide. The flasks are securely corked and left in the similar condition of light and temperature. A few days later, the seed in flask A will be found to be having clearly germinated, and those in flask B shows no sign of any germination. So of course, because they are dead, they are not going to germinate, but the flask A contains the germinating seeds, so they would have germinated because of the presence of water, oxygen as well as the temperature, warmth. The flask in each the gases in each flask are then tested by removal of the cork and tilting the flask over a test tube containing lime water, a dilute aqueous solution of calcium hydroxide, and then shaking up the test tube. The expected gas carbon dioxide being heavier than air would flow down into the test tube. The gas from flask A would turn the lime water milky, showing the presence of carbon dioxide in it, while the gas in flask B will show no effect. Therefore, the conclusion is that the germinating seeds give out carbon dioxide. The same thing can also be done in a different manner, जहाँ पे pour करने का झंझट नहीं करेंगे. उसके बदले what we do is we are going to take 4 flasks. So this is flask 1, this is flask 2, this is flask 3, and this is flask 4. Okay, you are going to connect it, so they are going to be sealed. Okay, and you are going to connect it this way, that there will be one tube entering inside, another tube coming outside, going into the next one, coming from here, going to the next one, coming from here and going outside over here like this. Okay, so the air is going to be made to enter over here, and here there will be lime water. So this is lime water, this again is lime water, here will be the seeds, and again lime water. So it's a simple process. What you can see now is the air enters here, goes into the flask. Because air has got carbon dioxide, this will turn milky. But when it is going to pass over here and comes here, this is going to not turn milky because it shows that carbon dioxide was no longer present in the air, which has entered here and is entering into the seeds. So we are sure that the gas, which is air, which is entering into this particular flask where seeds are there, contains only oxygen and nitrogen and no other gas. So this is no carbon dioxide list. There is no carbon dioxide which is there in the air over here. But you will find that after some time you will find that this lime water over here will turn milky. So kahan se aaya? Of course it is by the respiration of the seeds over here. So this is how you are going to have the experiment done. So a similarly more direct observable experiment to show the carbon dioxide produced during the respiration in germinating seeds is arranged in the figure as shown in figure. The air is drawn from flask A is cleared from any carbon dioxide present in it. The clear lime water in B confirms this thing. Confirms that the air entering into flask C is free from carbon dioxide, and the lime water in flask D turns milky, proving that the source of carbon dioxide was the germinating seeds. So that is how we are proving that carbon dioxide is going to be evolved during respiration. The next is experiment to prove that carbon dioxide is produced by green plants. So now in the same thing, we can have just a small change over here, that we will be having a plant here ओके एक प्लांट ले लिया, उसको एक बेल जार में डाल दिया. You're allowing the tube from here to enter in over here and tube from here to enter this over here. इसको निकाल देते हैं. So you see, air enters here. So air is entering into the flask. So यहाँ पे हम लोग ने क्या किया? We are taking over here soda lime. So we are taking, instead of having the bottle of one more flask, what we are doing is we are taking soda lime here. So soda lime, we know that it absorbs the carbon dioxide. So carbon dioxide will get absorbed over here. So here this is not going to turn milky. It is going to remain transparent. So it proves that there is no air, but no carbon dioxide present in the air which is entering into the plant. Whereas this is going to turn milky, proving that the plant produced carbon dioxide during respiration. So setup of an experiment is shown in 7.5 using a small potted plant such as germanium. The bell jar placed on should be placed on a glass sheet, and its rim as well as all other connections should be vesseled to make them airtight. Now this has to be airtight because अगर यहाँ पे air है तो यहाँ से अंदर ही air घुसेगा तो मतलब नहीं है कुछ उसका. So that's why, because it is necessary that we show over here that the air which is entering in the flask is free from carbon dioxide. But if there is a gap over here, then automatically वो कुछ मतलब का नहीं है. So this is the precaution what you need to take while during the experiment. Outside air is drawn into the apparatus with the help of an air pump. This air passes through the soda lime which absorb any carbon dioxide present in the incoming air, and the lime water A through which it passes will not turn milky. So this absorbs the carbon dioxide and then goes into this which will not turn milky, and then it passes over here into the bell jar. It is necessary that this experiment is carried out in dark. Now this is very important because these are not seeds. These is a plant. Now if it is a seed do not do photosynthesis, इसलिए problem नहीं है. पर ये plant है, plant photosynthesis करेगा, और अगर वो photosynthesis करेगा तो automatically if वो sunlight में है और photosynthesis करेगा तो it is going to give out oxygen and take in carbon dioxide. तो अपना जो पर्पस था ये कार्बन डाइऑक्साइड को चेक करने का, वो कार्बन डाइऑक्साइड खुद ही ले लेगा. It is going to take away that carbon dioxide for its own use. So then you will not get the results. So that's why this has to be done in dark, so that you are not allowing the plant to do photosynthesis. The experiment is carried out in dark, or the bell jar is completely covered with a black cloth to prevent photosynthesis, so that carbon dioxide liberated in the respiration is not used up for synthesis of starch. The next one is to show that heat is going to be evolved during germination. So here it's a simple experiment. You are just taking two thermos flasks, and the two thermos flasks are, it's just a simple thing. Thermos flask लिया, thermos flask क्यों लिया? So that you are not allowing the heat from outside to go inside and the heat from inside to go outside. So that is why we are using two thermos flasks. So this is one thermos flask. This is the second thermos flask. Of course the thermos flask is like this, so you will be having the insulation and everything over here. Okay, you are filling this completely with the seeds. So the seeds are put over here and seeds are put over here. Now of course these are dead and these are germinating. These are living, live. You place a cork and place a thermometer inside. So you're going to place a thermometer inside, and also here you are going to place a thermometer here. You will find that here there won't be any change in the temperature after some time, whereas here this is going to be giving you the change. So you see that take two thermos flasks and mark them A and B. Take about 30 grams of pea or bean seeds and soak them in water for about 24 hours. Divide the seeds into two equal parts. Kill one group of seeds by boiling them. and then wash them with dilute formalin or carbolic acid to prevent bacterial growth. Put the live germinating seeds in the flask A and killed ones in flask B. Insert a thermometer in each flask and plug their mouths with cotton wool. After a few hours, the thermometer in flask A will show a higher reading, thus indicating the germinating, that is live respiring seeds give out heat. The thermometer in flask B will show not show any rise in temperature. So a very easy example to demonstrate the heat which is generated during respiration. The last one is to prove that anaerobic respiration takes place in the plant, in the seeds. So let's see. Now for this particular thing, we'll be having only, okay, what we do is we are going to have one beaker containing mercury. Okay, there will be this beaker containing mercury, and then there will be a test tube. We'll take a test tube, एक test tube लिया, उसको पूरा mercury से भरेंगे, and then you put the seeds over there. Now mercury is heavier, seeds the seeds will float on this. So इसको फिर क्या करेंगे, ऐसा press करके उल्टा कर देंगे. So seeds being lighter is going to go up and settle on the top. So at the top you are going to get it like this, that the test tube will be ulta like this, with the seeds over here at the top and the mercury over here. So this will be the mercury and this will be the seed. So this is the seed and this is mercury. Now you leave this for some time, for a day or so, you will find that later on there will be an air gap in between over here. Okay, the seeds will be, of course, be floating here, and there will be a air gap formed at the top. If you check with a glowing splinter, you will find that that air gap what you are going to be getting, sorry, when you bring a KOH, yes, a potassium hydroxide to one side, and you introduce a potassium hydroxide inside, it will go up and absorb the carbon dioxide which is going to be there, so making it go upwards again. So it proves, because KOH ने carbon dioxide, KOH got the capacity to absorb carbon dioxide. So वो जैसे उसने ले लिया, it tells me that the gas which was present over here was carbon dioxide. So that means the seeds which were there had respired in absence of air anaerobically. So take 8 to 10 soaked and peeled pea seeds in a test tube. Fill the test tube with mercury and invert the test tube in a beaker filled with mercury. The seed will be completely surrounded by the mercury. After about 2 days, the level of mercury in the test tube will fall and the liberated gas will be found to be carbon dioxide. The carbon dioxide can be tested by introducing a stick of potassium hydroxide in the test tube, which will float above the mercury, through the mercury, and on coming into contact with the gas will absorb the carbon dioxide and the level of mercury will rise again. A similar control experiment could be set up in which the seed may be kept by kill them by heating or sterilizing them with some antiseptic. No gas will be liberated in the control. Note, peeling of the seeds coat of the germinating seed before introducing them in the test tube has hastened the outward diffusion of carbon dioxide. So we are peeling off the outer seed coat of the seed so that it increases the capacity of giving out carbon dioxide. Otherwise 2 दिन में तो मिलेगा नहीं और वो तब तक वो seed मर जाएगा and you will not be able to get the experiment properly done. So that's why you remove the seed coat and then do it for proper results. So this is the experiment which proves that the plants or the seeds are going to respire anaerobically. So yeah, this was about the chapter respiration. We already completed the later part, that is respiration, contrast of respiration and photosynthesis and contrast of respiration in plants and animals. We already done that in our previous video. So that comes to the end of the chapter respiration in plants. In one important thing over here is the structured application skill type chapter questions in the book back question, review questions में part E. Part E is structured application skill type questions. These are based on experiments and they are very very important because these are the kind of questions which they normally ask in exams for 5 marks. तो ऐसे ही होता है कि डायग्राम दिएंगे और वो डायग्राम के ऊपर there will be 5 questions. You need to answer the 5 questions and get 5 marks. So That's why it is going to be important for you to go through this particular page over here, that is page number 62 and 63 are the one which are structured questions. Okay, also continued in 64. So that was about respiration in plants. Thank you.